Okay, so thank you so much for the invitation to talk to you today. I have been following quite a few of the webinars in this series, and I think it's super inspiring and really, in a way, perfect for me to have these webinars right now, because my study is quite new, and I am gathering a lot of inspiration for what to look at in this study. So what I will talk about and what I am interested in this study is how the different adjuvant treatments, which is the treatments that these women with breast cancer receive after the surgery, so after the tumor is removed, they will get treatments to avoid breast cancer to come again and to metastasize to other tissues. And we are really interested in how this affects also the healthy cells of these women, and so in general the metabolic health and the other healthy cells of these women. So the aim of my presentation, which is also the outline, is that I will introduce you to our study called in Danish, Lev godt efter brystkræft. So unfortunately, we don't have an English title yet, but in general, this is have a good, like a good life after breast cancer. And then I will present some of the molecular data that we have so far. So we will look at how the mitochondria, so the energy metabolism, is affected by adjuvant treatment in both tissues and blood cells. And in the end, because my title has a bit of epigenetics in it, and we have unfortunately no epigenetic data yet, but I will present a few published studies on epigenetics and how epigenetics in different cells can affect metabolic health. So first, a little bit about me. So I work at the University of Copenhagen in this beautiful building, as you see in the picture, and I am a postdoc. I'm originally from Sweden, so just on the other side of Öresund, but I've been living in Copenhagen for 15 years now. And I was very lucky to receive one of the DDA postdoctoral grants to study both how breast cancer patients are affected by their treatment in relation to diabetes. I also did my PhD in Denmark, and that was in epigenetics and type 2 diabetes at the University Hospital, and also in a close collaboration with Linn University. And after that, I was getting my first postdoc in epigenetics and blood cancer, again at the University Hospital, but also at BRIC, the university, and I had a small visit at Van Andel Research Institute in Michigan in Peter Jones' laboratory. So in general, I try in this project to combine my knowledge in type 2 diabetes and in blood cancer to look at metabolism in women with breast cancer. And so this is a very translational study, so it's both involving clinical sciences and basic sciences. So how did it all start? Well, there was this guy, Mikael Andersson, who is an oncologist at the University Hospital. So he was a bit puzzled because he treats women with breast cancer, and he found that many of them got worse bone health. And so even if they treat the bone with aromatase inhibitors, in the case of postmenopausal women, some of these women cannot take it, like they get too many side effects. So he asked Professor Peter Schwartz, who is an endocrinologist working with bone health. So he asked, how can we do with these women? What can we give them instead? And so Peter did look into the literature. He couldn't find much on this, and he said, well, let's just study it. So he and at the same department was Charlotte Bruns, who was very interested in metabolism. So she was like, yeah, but why not also include metabolism and look at how they do after these treatments. So, and then Christian Buchlasen was involved as a PhD study. So he's the one recruiting these women. And then I came on the project to look at the molecular findings in both tissues and blood. And so Christian and Charlotte Bruns, they wrote this review where they looked at what is there yet on how chemotherapy and also endocrine treatment affects these women with focus on metabolism. And they found that there is a general trend that insulin increases and also insulin resistance increases. And this might be caused by weight gain or change in body composition, but it also might be caused by other mechanisms. So they started to include patients in 2019, and the inclusion criteria was that they should be postmenopausal women, aged 50 to 70, and they should have stage 1 to 3 breast cancer, which is early breast cancer, so it hasn't been metastasizing to other tissues. And they should be treated with adjuvant chemotherapy. And exclusion criteria was that they had already had a cancer or they had already endocrine disease such as diabetes. So this is our study design. So the women get their diagnosis, and at that time, Christian is contacted by Mikael Andersson to recruit these patients. He has around 5 minutes at these visits to ask them, do you want to participate in this study? And then they get their surgery, and before they receive chemotherapy, we take a blood sample, but not much more than that. And then we follow these women for 5 years. So after the chemotherapy, they visit us every 6 months or 1 year. And we measure anthropometrics, DEXA scanning, bone mineral density, and also metabolites and other factors important for metabolism. We also recruit a few of these women. We ask if they would be involved in an extra study where we do further investigations such as tracer meal tests, energy expenditure, and we also take fat and muscle biopsies. And we also include healthy controls that we can compare with. And later on, we will probably also investigate the same parameters during anti-estrogen treatment. And besides, we also look at neuropathy, vibration tests, heart rate variability, and we also have questionnaires. So we can follow how the women are doing also mentally. So, and we also have this app, maybe you have heard already about it. This is an app that every woman in Denmark with breast cancer can download and then fill out how they are, and in that way also contribute to science. So for the treatment, these women get chemotherapy, and of course it's not the same for everybody, but typically they get these 3 chemotherapies. And many of them also get radiation therapy for 25 to 45 treatments. They also get targeted treatments if they are HER2 positive, and all estrogen receptor positive women, they also get endocrine treatment with aromatase inhibitors. And to avoid osteoporosis, they get zoledronic acid treatment as well. And this aromatase inhibitors is given for 5 years, and zoledronic acid for 4 years. So far we have included 65, and unfortunately 18 are excluded due to Corona, especially because they have to go in an extra time to the hospital to take a fasting blood sample. So right now we have 47 active, and 36 have already been seen after chemotherapy, 25 after 6 months, 6 months after they receive aromatase inhibitors, and 13 also for 1 year after chemotherapy. So we don't have too much data to show you on the clinical side yet, but for the ones we have now, we see an average weight gain for around 3 kilos after chemotherapy. And this has been seen in other studies too, that actually around 3 to 5 kilos is typically seen after half-year chemo. We also see that the lipids in the plasma is a bit worsened. For example, HDL is dropping, and triglycerides seem to increase a bit. But we don't see any effects on glucose or insulin levels after chemo so far. So just as an overview, we know that adjuvant treatment of breast cancer affects metabolism, and the mechanism and the long-term clinical relevance remains unknown. So the overall aim of our study is to increase understanding of long-term consequences of this treatment from an endocrinological point of view. And also we want to investigate specific molecular mechanism of metabolic change. So this is the second part of my talk, where I'm going to focus on some of the molecular data, and especially on the mitochondrial respiration. So probably some of you are not familiar with this method. To measure how the mitochondria functions, you need to have a fresh sample of tissue or blood. So when we get the biopsies, we immediately go to the lab and we prepare the tissues. So we dissect them to have the cells separated and ready for respiration analysis. And for the blood samples, we isolate the peripheral blood mononuclear cells, and we do this by a density gradient. And then we take the blood cells or the tissue into a chamber of this oxygraph, and then we basically just measure how much oxygen we have in the media of these chambers, and how the tissues or how the cells, how much oxygen they use. And of course, we correct for the amount of cells. If there is a suspension, we correct for how many million cells we inject, or how much tissue we have in the chamber. And then we look at the oxygen consumption by the cells. And by doing that, we can actually study different OXPHOS complexes. And I will show you some data on complex 1 and 2, and also on the ATP production. So just going to show you some preliminary data. It's just on a few samples. And again, we have tissues after chemotherapy, and that's why we also include healthy controls to study how are women after chemotherapy related to healthy controls. And the first data I show you here is on muscle. So here we see complex 1 to the far left, and complex 1 and 2 in the middle, and then the electron transport system. So that's the maximal respiration of the tissue. And so to the left, the red points, that's the patients after chemotherapy, and to the right are the healthy controls. So it's kind of clear that the patients have a lower OXPHOS capacity in muscle tissue. But again, it's only based on 3 individuals in each group. And here in the bottom, we see the data we have so far on adipose tissue. And here we don't see any differences between the groups. But of course, we need more individuals to look at. And so, because we are interested in also what's happening with chemo in the same individuals, we thought of the idea to also include blood cells. So we asked the clinical biochemical department of the hospital if we could get an extra blood sample before the chemo, and we could. So since May last year, we analyzed how the blood cells respirate in these women before and after chemo. And I get a lot of great help from Ida and Lucas, 2 master students in the lab, who analyzed most of these samples. And we do this in both intact cells, so that's cells with a cell membrane intact. And then we also destroy the membrane of the cells to be able to add different substrates and see how the different complexes are affected. So first, some data on the intact blood cells. So here you see the oxygen flux, which is the respiration of the cells. So the basal is just the cells added to the chamber to see how the basal respiration is. And the ATP linked is how much oxygen they use for ATP production. And there is also a measurement of the maximal respiration. So to the far left, we have 19 patients included before chemotherapy. And we have 9 who also have been coming after chemotherapy. And then we have 6 healthy controls. And as you can see in this figure, there is no significant changes between the 3 groups. But if we zoom in on only the 9 individuals who we have data on before and after treatments, we can see a slight increase in the respiration. There is only a couple who decrease in respiration. And when I looked into the clinical data and the treatments they got, I found out that these few individuals who decrease, they did not get any radiation therapy. So if I exclude the ones who got only chemotherapy, we actually see a slight but significant increase in respiration of the blood cells. And this was nothing that we expected. And then the data on the permeabilized cells, here we see complex 1, complex 1 and 2, and ETS. We see again no significant differences between the 3 groups. And when we look at the 9 patients before and after chemo, they look like this. Again, just a few of them decrease respiration with chemo. And when we remove the ones getting only chemotherapy, there is a significant increase in respiration of these cells. So probably the radiation is doing something to the blood cells. Just to sum up, the chemotherapy might reduce the OXPHOS capacity in skeletal muscle, and still the sample size is very small. We do not see that the mitochondrial function in mononuclear cells is worsened. We actually see a slight increase in the patients receiving both chemo and radiation therapy. But of course, many other factors could influence the respiration of these tissues and blood cells. And then you could ask, what is actually the point of measuring blood cell respiration? Well, there is a few quite recent studies who indicate that blood cell respiration could reflect the bioenergetic state of other tissues. So it could be relevant it could be similar energy metabolism in blood cells as in other tissues. And that's why we are interested in this. And this is also something we will look more into. So before I end my presentation, I will talk a little bit about chemotherapy-induced DNA damage and epigenetic changes in blood cells and how that could be related to metabolism. And so there are a few studies showing that there might be DNA damage and reduced DNA repair in blood cells from women with breast cancer. And especially the radiation could induce these changes. So here we see also epigenetic changes. Actually, in this figure, there is a study where before and after chemotherapy, how the epigenetic age looks like. So this is something which is not a chronological age, but more a biological age. And they could see in this study that after the chemotherapy, it looked like that even if you corrected for the age in these women, that the epigenetic age was actually worsened. So you became a bit older biologically by the treatment. And this was especially seen in patients who only got radiation and not as much in the ones getting both chemotherapy and radiation. And there's also studies showing that mutations in blood cells might affect inflammation. And in this specific study, the age-related mutations was associated with coronary heart disease in humans. And in the figure to the right, the mutations of epigenetic regulator called TET2 was in animal studies associated with inflammation of the cardiovascular system. So this might also be true for other tissues, and that's how blood cells might be important to look into in relation to inflammation and metabolism. And also there are a few studies on macrophage infiltration, which are very important for, among other things, the regeneration of skeletal muscle in response to damage. So one could imagine that chemotherapy and radiation is causing damage to the muscle, and that we need to regenerate it. And here the macrophages might be very relevant to look into. Of course, it's also associated with like adipose tissue inflammation. So if we have a lot of infiltration of macrophages in adipose tissue, this might contribute to insulin resistance. So that could also be interesting, and this is also something we will look into in our study. So maybe the chemotherapy is altering both the tissues, but also the blood cells, and that this could be important for the development of metabolic disease. And so this is my last slide. So the main question was, how are healthy cells affected by adjuvant breast cancer treatment? And I think I showed you that both muscle, fat, and blood cells are affected by these treatments. The question is how much, and are these changes reversible? So will they actually disappear after half a year of after chemo? And this is something we would be very interested in looking into. And also, of course, all these treatments might also be somehow should be looked into with other eyes. For example, how are these women doing in regards to physical activity? How are they cognitive function? These are some factors we know already that affect these women a lot. And how are these factors related to metabolic health in combination with the treatments? So with these words, I would like to thank everyone involved in the study. And I would also like to thank all of you for listening, and the fundings, of course. And I'm open for any questions.
Thank you so much for this very great and inspiring talk, Linn. I really enjoyed it, and I will be chairing this session now. My name is Louise Lang-Lerskov, and I'm a DDA-funded postdoc. And if you have any questions, you can use the raise your hand function, as Jette said, or write your name and unmute yourself, ask the question, or you can write the question in the chat. And while people are preparing the questions, I have one for you, Linn. We discussed your study at an earlier time point, and at that time you were talking about having an exercise intervention as well. Yes. I remembered. Is that still the plan to include an exercise arm, or?
Yeah, so that's something we really want to do, because we are interested in also if exercise during the chemotherapy or after the chemotherapy could, of course, affect the health and metabolic health of these women. And it's been very hard to recruit these women. We have been trying very hard. Christian has been asking a lot of women, but the reason for why it's hard is because they just finished their chemotherapy and they are ready to start slowly going to work again, and it's not all of them who have enough energy for a different study. So some of them have been luckily willing to donate some tissues and participate in a one-day study, but to also doing exercise for many weeks is unfortunately not very easy to recruit.
No, it's difficult. Yeah, I could imagine. Yeah, and my thought was also if you do this exercise, maybe it would be a good idea to include diet registration, or you mentioned it as well, or a diet intervention. Maybe even it would be difficult, but we know that you're talking about this weight gain that they experience 3 to 5 kilos, and exercise training hasn't been shown to be able to reduce the weight. It induces, of course, insulin sensitivity is better, all these positive factors, but the weight gain in itself maybe will be difficult.
Yeah. So far we have the questionnaires, and so we ask a few questions about their physical activity, and we also ask some questions about their diets and mental health and cognitive function. So we have actually a few of these data, but only on a questionnaire basis.
Questionnaire. Exciting. Then we have a question from Amanda Leiter.
Thank you for such a great talk. That's a really interesting study. And I just had a question regarding steroid use in these patients. I'm not an oncologist, but I'm not sure if there's, high-dose glucocorticoids being given to manage side effects from the chemotherapy and how that may affect things in the study.
Yeah, that's a great question. Actually, I had the same question to Christian the other day, who recruits these patients. And typically, we don't give them any glucocorticoids. The oncologist, at least we don't have this data right now, but Christian will talk to the oncologist about this. But typically, no, not much of that. But it's a great question, and I'm sure it would affect the metabolism.